Although the syllabus was a kind of random, I have arranged in the serial wise by following BD Singh syllabus. So in today's lecture, in today's session, we shall see the first lecture, uh, Introduction to Genetics and Brief Historical Background. Then in next lecture, we shall discuss about the ultrastructure of cell. We know that cell is the structural and functional unit of organization and different cell organelles. What are the cell organelles? For example, mitochondria, ribosomes, chloroplast, lysosomes, and others, along with their functions. Next lecture will be the cell division. Cell division, two types, mitosis and meiosis. Mitosis, we know it is a kind of equational division where the chromosome numbers will remain constant from generation to generation. Whereas in meiosis, the chromosome number gets reduced to half. The stages and significance of these two cell divisions and difference between these two cell divisions. Next lecture about the chromosome structure, its morphology, and about the karyotype and ideogram, difference between these two concepts. Then chromosomal aberrations. We know the normally the chromosome structure and its number, if it gets disrupted, then we call it as chromosomal aberrations. These aberrations are of two types, structural and numerical. Structural chromosomal aberrations, these lead to change in the overall structure of a chromosome by addition, deletion, or inversion or translocation of a gene. Whereas in numerical chromosomal aberrations, the chromosome number or whole set of chromosomes will get changed. If whole set or the genome is getting doubled or tripled or by many numbers, we call it as euploidy. And if only one or two chromosomes are getting changed, then we say it as aneuploidy. We will be taking examples of wheat, tobacco, cotton, and brassica. And from here, we shall start the transmission genetics, that is Mendel's law of inheritance. Mendel, he is the father of genetics, Gregor Johann Mendel. He has given two laws of inheritance, law of segregation and law of independent assortment. We shall discuss these two laws in detail and what are the exceptions to these laws. And then cytoplasmic inheritance means inheritance due to presence of genes in the cytoplasm. In cytoplasm, where the genes will be present, especially in mitochondria and chloroplast. So the mechanism of cytoplasmic inheritance, its characteristic features, and difference between the nuclear and cytoplasmic inheritance we shall study. Then what is maternal effect? Then in next lecture, we shall see the multiple alleles. Means in general, a gene will be having two alleles, one dominant and one recessive. In case of multiple alleles, there will be more than one alleles for a particular gene. That phenomena, we call it as multiple alleles. Classic example of this multiple allele is, for example, human blood group ABO. Then pleiotropism, in which one gene is responsible for so many characters. Then a phenomena related to the expression of a particular character. Those are penetrance and expressivity. Then in next lectures, we shall see the gene actions, two types of gene actions, intragenic and intergenic. In intragenic gene actions, three types are there: complete, incomplete or partial dominance, and codominance. Then in intergenic gene actions, all type of digenic interactions, like first is dihybrid ratio given by Mendel, and then complementary, supplementary, inhibitory, masking, and polymeric, additive, and many others, along with the examples. And the next lecture will be the linkage and crossing over. Linkage is nothing but the capacity of two or more genes to stay together during inheritance. Why they will stay together during inheritance? Because they are located on the same chromosome. And crossing over, it is a phenomena which leads to variation in the progeny. So linkage, its types, detection, estimation, and exploitation. Then sex linkage, sex limited and sex influenced traits, different mechanisms of sex determination with examples. And in mechanisms, we shall try to study these sex determination mechanisms in plants and animals separately. Then crossing over mechanisms and the factors which affect the crossing over, means which increase or decrease the frequency or occurrence of crossing over. We shall try to study those. Then next lecture about the introduction to quantitative genetics. Two types of traits, qualitative and quantitative, and difference between these two types of traits. Then multiple factor hypothesis, population genetics, and Hardy-Weinberg law. And in next lecture, mutation. Mutation is nothing but a sudden heritable change in the phenotype of an organism. So what are the characteristic features of these mutations? And mutagens, we shall study the different types of mutagens, their classification with examples. Here in syllabus only, they have clearly mentioned to omit this CLB technique of detection of mutation. So we shall see only different techniques through which mutation can be detected. Then DNA, until here, we will be discussing about transmission genetics and all. From now on, the molecular genetics part will start after this. That is, DNA means genetic material, and the structure of genetic material, how this genetic material is replicated and all. So in this lecture, DNA, its structure, function, types. RNA, its structure, function, and types, along with the mode of replication. We know how DNA is replicated. In which mode DNA gets replicated? Do anyone know?
Transcription.
No, whether it is semi-conservative, conservative, or dispersive.
Semi-conservative.
Yeah, it's correct. So we shall see the different modes of replications. Then the basic phenomena in molecular biology, which has been given by Francis H. Crick, called as central dogma, in which we shall see how the DNA, it gets from the DNA, mRNA is formed, and how from mRNA proteins are formed. When DNA gets replicated, it only forms DNA. But through the method of transcription, from DNA, mRNA is formed, and through translation, from mRNA, proteins are formed. This basic flow of information from the DNA in the form of mRNA to the proteins, and these proteins, when they work, they lead to expression of a particular phenotype. For example, hair color, skin color, etc. So this basic flow of information in an organism is called as central dogma. We shall try to study in detail, means steps involved, and RNA polymerase, DNA polymerase involved in these mechanisms. And the genetic code as well. Is it triplet genetic code, or with 2 amino acids only it is there, or 1 amino acid?
Triplet, no?
Yeah, okay. Then in next lecture, we shall study the gene regulation, which means that how a particular gene produces its expression or phenotypic characters. Here in syllabus, they have mentioned to take Lac-Operon as a model. So we shall study the Lac-Operon model. Until here, all the basic syllabus of genetics will be covered according to B.D. Singh. And in later, they have given the practical aspect of genetics as well, like microscopy and preparation of slides and all. That we shall try to discuss in a separate class about microscopy, light and microscope, and electron microscopes. Then preparation and use of fixatives and some stains for light microscopy. Here, for preparation of slides and identification of various stages of mitosis and meiosis, we will be covering in the cell division chapter only by seeing different phases of chromosome structures in different stages in mitosis and meiosis. And in last part of genetics, we will be solving the numerical problems of monohybrid ratio, dihybrid ratio. We know that monohybrid ratio is monohybrid cross. It involves only one gene when only for one gene cross is made, and or one character we can say. When for two characters the cross is made, it is dihybrid. So numericals on these basic concepts of Mendel. Along with that, some statistical testing of segregation of traits as per Mendelian principles, chi-square test, its application, chi-square, we will be discussing it in statistics as well in our BSc, right? So by taking genetics examples, we shall try to discuss this chi-square test. And then digenic interactions means those only, like complementary, supplementary, inhibitory, all types of gene interactions. And polygenic inheritance, gene mapping, autosomic and sex linkage. This will complete the genetics part. And from next lectures, we shall start the plant breeding. From here, first lecture of plant breeding will be importance, objectives of plant breeding, centers of origin, especially given by Babilo. Then plant genetic resources, their conservation, utilization in crop improvement, and domestication, which is nothing but bringing of the wild species under the human management. Then concept of plant introduction, again primary and secondary introduction, and acclimatization. In next lecture, the modes of reproduction will be studied. What are the modes of reproduction? They are asexual and sexual. In asexual, again, two modes of replications are there, vegetative and apomixis. So we shall try to discuss these things in detail and what are the genetic consequences of these modes of reproduction, cultivar options, and their significance in plant breeding. Then modes of pollination. There are two modes of pollination. What are those two modes of pollination?
Self-pollination, cross-pollination.
Okay, and
From
another
cross-pollination.
intermediate between these two, when it is approximately, it reaches, the cross-pollination reaches 30% in self-pollinated crops, it is regarded as often cross-pollinated. So genetic consequences of these modes of pollination and difference between the self and cross-pollinated crops. Then objectives and concepts of breeding self-pollinated crops, cross-pollinated crops, and vegetatively propagated crops. Okay, can you give an example of a crop which is vegetatively propagated commercially?
Banana.
Okay, right. Then in next lecture, the pollination control systems means to control the pollination, two basic systems which are genetically, we can say self incompatibility and male sterility, and their utilization in plant breeding. We shall see the basic mechanisms, types of self incompatibility and male sterility, and how these are being utilized in plant breeding. How they can be utilized means especially for the production of hybrid seeds, we know. And then idiotype concept, and means the model will be given for a particular crop so as to maintain the overall high yield with less inputs required and everything. The different models for specific crops will be prepared with reference to grain number, plant height, and other genetic characters. And then introduction to IPR. IPR means intellectual property rights. Then emasculation means removal of male part or anthers especially. Then hybridization techniques in crop plants, methods of generating variability, hybridization, mutation. What are those methods of generating variability? Those are hybridization, then mutation, polyploidy, soma clonal variation, then mass selection, pure line, Johnson's pure line selection, and genetic basis of this pure line selection. Why just having a glance on syllabus is important? Here we can see that here they only mentioned Johnson's pure line selection. And after this, seeing the brief outline of syllabus, we shall see some of the previous year questions which have been asked in 2017 paper. There one question directly came that who gave this pure line theory? In syllabus only if we see, answer is there. That's why at least before starting reading for any subject, we shall have a small glance on our syllabus. Next lecture will be method of handling the segregating generations. First, we will create this variability, and then how to handle these segregating generations in self-pollinated crops like pedigree method, bulk method, single seed descent method, modified methods of these all, and then backcross method and its applications. Then concepts of population improvement like mass selection, modified mass selection, recurrent selection methods, and development of synthetics and composites. The next lecture will be concepts of inbreeding depression and heterosis, Here we shall see different theories of given for heterosis concepts, then utilization of these heterosis for development of hybrids in like for inbred lines, single cross and double cross hybrids. Next lecture will be principles of breeding clonally propagated crops, concepts of handling the breeding material generated through mutations, through polyploidization, and significance of wide hybridization. It is also called as distant hybridization, means when a particular crop is crossed with another species or another genera. That is called as wide or distant hybridization. Then related to some aspects of biotechnology for crop improvement, especially the tissue culture, anther culture, doubled haploids, which are especially used commercially in maize, then embryo culture, soma clonal variations, and in next lecture about introduction to markers, different types of markers and marker assisted selection. In next lecture about origin, evolution, distribution of cultivated and wild species in some of the cereals, pulses, and oilseeds, and fibers, and commercial crops. Only these many crops have been mentioned. For example, in rice, in cereals, rice, wheat, maize, sorghum, bajra, ragi, and in pulses, red gram, green gram, black gram, soybean. Soybean is also an oilseed. Then the groundnut, sesame, sunflower, castor, and brassica. In fibers, only cotton, and in commercial crops, sugarcane. Then next lecture will be for biotic and abiotic stresses, means breeding for resistance to these stresses and breeding for quality traits as well. Quality trait means like not only yield, but for example, the protein content or reducing the anti-nutritional factors. This all comes under the breeding for quality traits. Then mechanisms of resistance and selection criteria for resistance to abiotic and biotic resistance stresses. This is also a practical aspect of plant breeding which has been given in syllabus, like visit to research centers of different crops. Here we shall try to list all the research centers of different crops throughout the country. Then visit to GPB breeding plots and demonstration of segregating generations, then centers of different steps crops, and here steps of variety release we shall try to see in state as well as in country. If we want to release a variety, what are the steps to be followed, including the dust test. Dust test means distinctiveness, uniformity, and stability. These three characters must be present in a variety to be released. So then field layout of experiments, percentage of outstanding recently released varieties. See, parentage, I mean, whatever all the released commercial varieties are there, by taking which parents these varieties have been prepared, we shall try to remember these things. And then quality characters, sources of donors, not only parents. If for example, for breeding for biotic and abiotic stresses, some of the donors will be used. So which specific donors were used for producing which specific variety, we shall try to remember those things. Visit to seed production and certification plots and AICRP programs for different crops, and grow out test plots, seed production plots, and other institutions. If we talk about general weightage which we can expect for GPB subject, after analyzing the paper of 2017, for GPB, around 8 to 10 questions were there. And if we combine it with seed science and technology, approximately 12 questions were there. It means in plant breeding, we will be reading the basic aspects for production of different, like for example, hybrid seeds. For those things, the practically for each and every crop in seed science and technology, it will be dealt. So if we combine the GPB and seed tech, approximately 12 questions can be asked if they follow, the pattern will be followed as per the 2017 paper. Here we shall try to see the different questions which have been asked in the 2017 papers. These are the questions. These are the questions taken from the 2017 paper of AAO. First question, it consists of GPB as well as CTEC questions. First question was minimum germination percentage of certified seed in hybrid maize. Certified seed, we know how many classes of seeds are there: nucleus, breeder, foundation, and certified. So for certified seed,
90%.
yeah, it's 90%. In general, in maize, 80% will be there. But for certified and hybrid seed of maize, it will be 90%. Next question was about hybrid seed production of A and B lines. A and B lines in hybrid
Isogenic ones.
Yeah. Why they are isogenic? We know that in hybrid seed production, 3 lines will be used, 3 different lines. These will not be of different crops, but same crops, but they will be having one gene difference of male sterility. It will be male fertile and it will be a restorer. A and B, these 2 lines will be almost similar for all the genes, except that A will be sterile and B will be fertile. So that B maintains the A line. That's why A and B are isogenic lines.
Pure line theory, Johnson.
Okay, we saw just now in syllabus Johnson's
Johnson.
pure line theory. Oh, yeah. Then what was the contribution of Watson and Crick?
Double helix
Double
model.
helical structure.
Double dogma.
Double helix
DNA.
model of DNA. Yes. And Stadler, it was mutation. After Muller, he carried out mutation experiments using X-rays in plants. Then Beadle and Tatum in bacterial
Gene one
genetics.
enzyme hypothesis.
Okay, good. Then the next question was recombination frequency between linked genes. Here we know that linkages means if 2 genes are linked, they will be transferred to progeny as it is. They won't change their position. But if recombination occurs, what will the frequency of these genes to the maximum? It will be just half of the crossover frequency. Crossover frequency means crossing over, it will be occurring very less in the linked genes. That's why to the maximum, only half of the crossing over frequency, the recombination frequency can occur. Okay, then next question was each artificial chromosome vector can accommodate about what kind of, like how much base pairs of foreign DNA?
1 MB, ma'am.
Okay. What about bacterial artificial chromosome? Do anyone know how much it can carry? Means different types of vectors will be there, like plasmids, then YAC means yeast artificial chromosome, then bacterial artificial chromosomes. So BAC means bacterial artificial chromosome. It can carry up to 300 to 350 kilo base pairs. Means YAC means yeast artificial chromosome is having the highest capacity to take large size of fragment of DNA as a vector. And then stratification of seed is carried out by D, placing seeds in layers of sand at low temperature. Means stratification is done. Why it is done? Stratification. Do anyone knows? To break the dormancy. So in stratification, the different these seeds, they will be placed in layers of sand at very low temperature. Then genetic structure of a pure line.
Homozygous,
line?
homogeneous.
Okay, right. Homozygous, homogeneous. What is pure line? What is pure line? It is the progeny of a single self-pollinated homozygous plant. That's why it will always be homozygous as well as homogeneous. Whereas the genetic constitution of a landrace variety of a strictly self-pollinated crop. See, here they are saying landrace variety also and strictly self-pollinated variety also, crop also. For any crop, what is the answer?
Is it homozygous, heterogeneous?
Yes, it is correct. Why? Because strictly self-pollinated crops means we shall understand here that any crop which is strictly self-pollinated, it will always be homozygous. And as this is a landrace variety means these are the dynamic populations which have been evolved by farmers' efforts by local breeding. That's why they will be heterogeneous. The landrace variety of a strictly self-pollinated crop will be homozygous and heterogeneous. They are the varieties which have been bred by earlier farmers, not the by modern breeding methods. That's why they will be heterogeneous. Then next question was DNA fragment to be introduced into the host cell.
Desired DNA.
Okay, desired DNA or desired gene. Then what is primer? Especially in genetics, if we are solving MCQs, we should not be focused only on correct answer or correct multiple choice. We shall try to understand about remaining 3 options as well. Then it will be a kind of good revision. So what is primer? Anyone knows? Primer, it is a short synthetic single-stranded DNA or RNA, which is a kind of starting point for DNA synthesis. Then intron, we know introns and exons, especially in eukaryotic genes. These introns and exons will be there. That means discontinuous genes will be there. Introns, they will be removed after mRNA splicing, because in eukaryotes, when DNA is transcripted to form mRNA, when DNA is transcripted to form mRNA will be having the introns, the base pairs of introns as well. After splicing, these non-coding the base pairs or genes will be removed through a process called splicing. Then it will be only the coding genes which will be present in the mRNA. Those are called as introns. Desired DNA, we know desired gene which is about to be introduced into the host cell. Headquarters of ICRISAT, where it is
Hyderabad.
located?
Hyderabad.
Yes. First transgenic cotton variety approved for commercial cultivation in India. In which year?
2002.
2002, ma'am.
March
Okay.
2000.
Yes, you are well prepared. It's 2002. What was this transgenic cotton variety, for what it was produced?
Bollworm resistant.
Bollworm resistant.
Okay, it was BT cotton, that is Bacillus thuringiensis, for bollworms resistance, American bollworms. A method of handling segregating generations when there is maximum opportunity for a breeder selection is?
Pedigree method.
Okay. Why it is pedigree method? Because in pedigree method, the breeder will be having the data about all the previous generations of that particular line or breeding line. That's why he can analyze for each and every generation how the crop performance was there and how its pattern is getting changed. That's why with all the prior information with different generations, the breeder can select maximum opportunity in pedigree method. In bulk method, like all the seeds, they will be bulk and just forwarded to the next generation. In single seed descent method, although breeder can select, but it can select only one seed per plant. That's why the chances will be there that most of the desired seeds can also be eliminated. Then in mass selection, at a time with phenotypic characters, more than one plants will be selected for a particular generation. Then minimum number of field counts to be taken in an area of 10 hectare. Sorghum seed production RC. Again, this is a question from seed production. So how many counts should be taken for 10 hectare area in sorghum crop?
Ma'am, is it 9?
Yes, it's 9. If we see, like for almost for 10 hectares, 9 counts should be taken. For 6 to 8 hectares, 8 counts should be taken. And for 4 to 6 hectares, approximately 7 counts should be taken. Why these counts should be taken? What are these counts? These are nothing but the field inspections to carry out Logging, we know that off types will be removed, like those which are genetically not pure will be removed, and overall the hygienic conditions of that field, will, like cultural operations, presence of weeds, diseases, and pests will be analyzed. So for that, field counts will be kept, field visits will be kept. So for 10-hectare area of sorghum, at minimum, 9 field counts should be taken. Then maximum number recombination between two linked genes is
50%.
okay. It is 50%, because if the two genes are linked, the recombination between them cannot cross above the 50%. That is 50% only. Right answer. Okay. These were all the questions which have been asked in the previous years. Here we can see that only one question. Maybe we can take this question as genetics also and biotechnology also, because both of their syllabus are overlapping for some extent. If we take account, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. See, approximately 40 questions were there, including genetics, plant breeding, seed tech, and one question of biotech. So we have discussed all the previous year questions. Now we shall start our first lecture. Hope every question is clear here. All the previous year questions. Any doubt in these questions? Okay.
Ma'am, we had solved earlier, ma'am, so I could answer, ma'am. But we don't know each in particular.
Yeah, in while discussing the syllabus, like in every lecture, we shall be studying in detail. So you can get to know about the basic concepts, and you will be able to solve the questions without reading as well.
Ma'am, by when syllabus gets completed, ma'am? Can we expect this to be completed by end of March, ma'am?
Okay, for genetics, I can complete within 10 days. For plant breeding, it will take some time, it seems. For genetics, I'll try to complete it soon. If you all are ready, means I'll take three classes also for a day, or 4 classes. If your classes schedule is not busy, then if it is busy, then we shall try to complete in 10-15 days. For genetics, I'm saying.
Okay, ma'am.
Teach little slowly, ma'am.
Okay. So now we shall start the second lecture, means first chapter of genetics, introduction to genetics, brief historical background.
Ma'am, your introduction, please.
I am from genetics and plant breeding department only, but every details will be kept confidential for some reasons. Sir has already mentioned, I think, before starting pathology class. So I am this faculty of genetics and plant breeding only. Don't worry, I am not from agronomy and pathology. Okay.
Extension was taken by you.
No. I didn't took extension.
Okay, ma'am. Okay.
Okay. So we shall start the second lecture about introduction to genetics. Before starting, we should analyze here that a keen observer, he will readily recognize two basic aspects of this biological world. What are those two basic aspects? Are that progeny of an organism will develop similar characteristics to that organism, and another is that the variations, means some of the differences will be present among the individuals of a single species. Means that no two human beings are identical to each other in every aspect. That is called as variation. And this the phenomena where the progeny of an organism is having some of the similar characteristic to that species or organism, we call it as inheritance. So by analyzing this, we can say that genetics, it is the, if I am getting fast, let me know, I'll slow down my speed. Genetics, it is the biological science which deals with the mechanism of heredity and variation in the living beings. Heredity, it refers to the transmission of genetic information, more precisely the genes, from parents to offsprings. What are transmitted from parents to offspring is the genes, and variation refers to the difference among the individuals of a species for a character. The word genetics is derived from the Greek word. From which word? It is derived from the Greek word genetikos or genesis, which means to generate or to become or to grow. The father, we know father of genetics is Gregor Johann Mendel, but the term genetics was coined after the almost 25 years of his death by William Bateson. Who coined the term genetics? William Bateson in the year 1906. He was the one who translated Mendel's papers from German to English. Then scope of genetics, it is essential to understand the other fields of biology as well, and it has socio-economics contribution for human welfare, such as improvement of human race, eugenics, for crop improvement, for some of the medical applications, criminology and legitimacy, means for making the rules and regulations, then for animal improvement. To study the genetics, we can overall we can divide this genetics into three different branches. Modern genetics is traditionally classified into three branches as transmission genetics, molecular genetics, and population genetics. Transmission genetics. What is transmission genetics? It deals with the study of passage of characters from one generation to the next generation. It is studied mainly through breeding experiments in individual organisms. Transmission means transfer of characters or genes from one generation to next generation will be studied in transmission genetics. It also encompasses the basic principles of genetics, such as relationship between the chromosomes and heredity, arrangement of genes on chromosome, and gene mapping. It has important practical applications in the development of new varieties in plants and animals. As Mendel pioneered this approach to genetics, it is frequently called as Mendelian genetics or classical genetics. Transmission genetics is also called as Mendelian genetics or classical genetics because Mendel was the first to carry out first correct experiments on garden pea to study the inheritance and variations. He carried out his experiments for approximately 7 years. Then next branch, second branch is molecular genetics. It involves the study of chemical nature of gene, its structure, organization, function at the molecular level. It includes the cellular processes such as replication, transcription, and translation, and also the gene regulation part, which is the process of control of expression of the genetic information. The developments in molecular genetics has led breakthrough in cloning and genetic engineering as well. If we are reading the gene, its chemical nature, its structure, organization, and function at molecular level, it is molecular genetics. Then next, third branch is population genetics, which means the study of variation of genes between and within the populations. We are not studying at individual plant level here. We are studying at population level. This the population of a particular species and its changes over time and space. It is important in evolutionary studies as well, as evolution is just the change in genetic makeup of population over time. However, these fields may overlap each other and each organism, for example, Drosophila or maize, may be studied at any level of genetics. It will be up to our objective of study with which branch of genetics we will be dealing with. There are two concepts of gene, more classical concept and modern concept. Classical concept of gene. We know that before the Mendel gave his conclusions about the gene, that genes are the discrete particles, some of the believers, what they were believing is that the genes or the hereditary material might be blood or any kind of liquid. It was only after the Mendel's experiment that we got to know it clearly that genes are the discrete particles which occupy specific loci. They are present in specific location and responsible for expression of specific characters. Then genes are arranged in single linear order on chromosomes. beads on a string. Like beads on a string, we shall study this in detail, how these genes are arranged on chromosomes. Then number of genes is always more than a number of chromosome, because in one chromosome always more than one number of genes are present when we are talking about the eukaryotes. Any doubt? And genes are the units of heredity, means these are only the units which will be transferred from the parents to the progenies. And genes are capable of self duplication, producing their own exact copies, and they may undergo sudden changes in its composition and position, which is called as mutation. This was the classical concept of genetics. Coming to the modern concept, although the definition of gene has been changed over time with the discovery of then DNA, protein, and enzymes, everything, but modern concept of this gene was given by Seymour Benzer in 1957. He coined the term for describing genes. He coined three terms for describing the genes. In three terms, what are those? Cistron, recon, and muton. Cistron are the units of transmission. They may be regarded as synonym of a gene, means which gets transmits from generation to generation. Recon, these are the unit of recombination. And muton, these are the units of mutation. So first term for to describe gene is cistron. It is the unit of transmission, a segment of DNA which codes for one polypeptide or RNA. Generally, it is accepted as synonym of gene, which encodes for one polypeptide or RNA. This is important. What is the function of this cistron? Means one polypeptide or RNA it produces from the DNA. Then what is recon? It is the unit of recombination, the part of gene within which recombination cannot take place, represented by one nucleotide pair. One nucleotide pair means it may be AT or GC. Why recombination cannot take place between these two? Because there must be some adjoining base pairs within them, beside them, so as to recombination or crossing over to take place. That's why recon is just one nucleotide pair within which recombination cannot occur. And muton, it is the unit of mutation, the smallest unit of gene capable of undergoing mutation. It is represented by one nucleotide pair as well. See, here in muton, it can undergo mutation, like in place of A, in place of T, again G might come, C might come, and if it is in DNA, in RNA, U might come in place of A, adenine. So within one nucleotide pair also mutation can occur. That one nucleotide pair, which is the smallest unit of gene, which can undergo mutation, is called as muton. So some of the genetic terms we should be knowing
Hello.
to understand the.
Hello, ma'am. Hello. ಮ್ಯಾಮ್ ಅದು ರಿಕಾನ್ ಇನ್ನೊಮ್ಮೆ ಹೇಳಿ ಮ್ಯಾಮ್.
Recon. Okay. See, recon is a unit of recombination. Recombination means we know after crossing over, recombination happens. The part of gene within which recombination cannot take place. We are here, like for in mutation, we are saying that mutation can occur, but in recon, within which recombination cannot take place. Why it cannot take place? For recombination to occur, some adjacent genes or nucleotide pairs, for example, for genes, adjacent genes should be there to crossing over. Similarly, for recombination to occur, if we say in terms of base pairs, in DNA, what will be the base pairing we know? For example, A pairs with T, G pairs with C, right? So here recon is getting represented by one nucleotide pair, means only this much is recon. This is not there, means if we say only AT. How recombination can take place just between these two? for recombination to take place, at least here some other GC or CG must be present so as to get interexchanged. But we are considering recon only as one nucleotide pair, nothing else beside them at any side. So there won't be any chance of getting interexchange of in just one nucleotide pair. That's why in recon, recombination cannot take place. In more than one nucleotide base pairs, we can expect recombination to occur. Okay. To understand
Okay,
this,
ma'am. Thank you.
to understand this unit of recon, that is recombination, we shall know that for anything to cross over, at least two things should be present. If we are considering the unit only as one, then we can't make it interexchange. That's why the recon is the unit which under which recombination cannot take place. Hope it is clear now. Then we shall move on to genetic terms. Some of the basic terms to understand the other aspects of genetics. For example, genome. The sum total of all the genes present in an organism are genome. And locus, it is a physical location of a gene on a chromosome. Or we can say the particular address of a gene on a chromosome. Allele or allelomorph, it is the alternate form of a gene. Alternate form of a gene in the sense, for example, a gene governing the height or plant height, we can say it is having two alleles, one dominant and one recessive. Dominant will govern tallness and recessive will govern dwarfness. These two different alternate forms of these genes, when present with some of the variations, it might produce tallness, it might produce dwarfness. These two alternate forms are called as alleles or allelomorphs. Then genotype and phenotype. Genotype, it is the genetic constitution or genetic makeup of an organism. And phenotype, it is the observable characteristic feature of an organism, which we can see morphologically with our own eyes, that is phenotype. And the actual genetic constitution behind this phenotype is the genotype. And the concept of homozygous and heterozygous. Homozygous, heterozygous. For as just now I said, for example, for plant height, a gene is having either capital T or small t. Means these are the two alleles. If the for a particular plant is having the alleles only for tallness or only for dwarfness, then this condition means presence of two similar alleles will be regarded as homozygous. And when the alleles will be different at two different loci, we'll be calling it as heterozygous. This is like a genetics background student can easily understand if I say that two alleles will be there. But practically, if we want to understand the the actual how these alternate alleles will be present, I will give a small example here. We know that humans are having how many chromosomes?
46.
Okay. So in these 46, 23 comes from father, 23 comes from mother. And these chromosomes will be present in pairs, right? Like pairs will be there, like these 23 pairs will be there. So if we say person as homozygous for a particular gene, when in this chromosome as well as for example, if this chromosome is governing a particular trait and this chromosome is having similar allele, small t only, then it is homozygous. If one chromosome which came from father is having capital T and another chromosome which came from mother is having small t, then it is heterozygous. Hope it is clear now how the alternate forms of genes that are alleles present in a particular organism. Then dominant and recessive traits.
Ma'am, homogeneous.
Yeah, homogeneous, heterogeneous is another concept which is related to we can say at the population level. Homogeneous, heterogeneous, we can't say at individual singles at individual level. Means we can't say a particular single plant as homogeneous, because at the population level, if we see that so many plants are there, and they are all of similar genetic constitutions, then that population is called as homogeneous. Whereas if we are studying the plant, if we are studying the single plant, and then we can say whether it is having two similar kind of alleles or two different kind of alleles. Then single individual level homo or heterozygous will be present. If we are observing the whole population, then we can say it as homogeneous or heterogeneous, means whether all plants are having similar genetic constitution or not. Understood?
Yes, ma'am.
Okay. Dominant and recessive trait. The trait is expressed when organism inherits at least one dominant allele, and recessive trait means it is expressed only when an organism inherits two alleles, two recessive alleles. Why? For a recessive allele to express only two alleles should be present, and why for dominant at least one? If one dominant allele is present, it can express, because if we see for a gene T and small t, if both capital T or small t are present, this produces tall, this produces dwarf. But in case of heterozygous condition, whatever these, it will be present on separate chromosome, it will be present on separate chromosome, right? So capital T, whatever expression it produces, it can mask, or like it can overcome, or it can suppress the expression of this small t allele. That's why the whatever phenotype T was going to produce will get hidden by capital T, means the plant will produce tallness. That's why the dominant trait, which is expressed an organism inherits at least one dominant allele. If at least small capital T, one capital T is there, it will produce small tallness and suppresses the dwarfness nature, which was going to be produced by small t allele. But as the small t functions are getting suppressed by the capital T, so when it can express genuinely its expression, like its phenotype, only when T will be absent with it, only when small t will be in homozygous conditions, then only the tallness, dwarfness will be produced. So dominant trait is expressed when an organism inherits at least one of the dominant allele, and recessive trait is expressed only when organism inherits two recessive alleles. This was about the two types of traits. Then about the other two kinds of alleles, wild type and mutant alleles. The most common allele in a population and mutation in wild type allele produces a mutant allele. In overall population of a particular species, if we observe, whichever will be the most commonly occurring allele will be regarded as wild type, and over the time when some mutation occurs and it leads to some small changes in that allele, it will produce mutant allele. These were some of the terms. Not only these, some other terms are also there, which we shall see wherever they comes later in the different lectures. Now we shall see that about the forward and reverse genetics. Generally, forward genetics, generally what we see, what Mendel did, he observed that a certain pea plant was producing either tallness or dwarfness. This was the phenotypic expression he was seeing. Later, some other scientists worked about the molecular aspect of those particular traits or genes, and they got to know actually which gene is responsible for that trait and which protein was produced by that gene, and what is the actual DNA sequence of that gene. Means if we are going from the phenotypes to the gene, protein, and DNA sequence, it is called as forward genetics. And in reverse genetics, we will be having a known base sequence of genes. Some of the base pairs of DNA we will be having, and we will decode this protein, this sequence, which protein it produces, and what functions this protein does, and through which which phenotypic trait is expressed. Means if we are going from the molecular level to the phenotypic or morphological level, we can say it as reverse genetics. So forward genetics and reverse genetics. Hope it is clear. Next, Gregor Johann Mendel. He was born in 1822 and he died in 1884. He is called as father of genetics. He was a Moravian Augustinian monk in 1850s. He worked with tea plants, Physarum sativum, for 7 years. What he studied? He studied the inheritance of 7 contrasting characters of tea. His paper, Experiments in Plant Hybridization, was published. Who published this paper? Experiments in Plant Hybridization means in English, Bates and did. Whatever he published in 1866 was in German language, called as Versuch über Pflanzenhybriden. Where he published this paper? It was published in volume 4 of Annual Proceedings of Natural History Society of Brunn. Later about Mendel, we shall see in detail in the Mendelism chapter. So here in genetics, we will study about the cells, cell organelles, and cell organelles and chromosomes. So study of cell and different cell organelles and their functions. If we are studying the structure, function, and molecular organization of cells, it is called as cytology. It is derived from a Greek word. Cytos means hollow vessel and logos means to discourse or to study. Therefore, cell biology is the biological science which deals with study of structure, function, molecular organization, growth, reproduction of the cells. And cytogenetics, it is the branch. If we are studying in a cell, we know so many cell organelles will be there, nucleus will be there, inside the nucleus chromosomes will be there. So if we are studying the behavior of these chromosomes in relation to the heredity, then it is known as cytogenetics. You don't get confused between cytology and cytogenetics. Cyto means simply cell and logy means simply study. So study of overall cell and its organelles and functions. Cytogenetics means we are going through the genetics aspects as well. Genetics means inheritance. Inheritance means what will get inherited? Chromosome. Therefore, the branch of genetics that correlates the structure, number, and behavior of chromosomes with heredity and variation is known as cytogenetics, or the study of chromosome, their effects on the development of character of an organism. Cytology, about cell and cell organelles. Cytogenetics, chromosome in relation to heredity. Then here are some of the model organisms which we take for studying in genetics. What are these model organisms? We shall see. In case of bacteria, we mostly do research in E. coli, and we also use bacteriophages in insects, mostly on Drosophila. In plants, mostly Arabidopsis will be used, but maize, due to ease in some genetic aspects, maize is also used, and in animals, we conduct experiments on mice. Okay, so here we shall see some of the advantages or benefits of taking the specific organisms as model organisms. For example, E. coli, it is having generation time of only 20 minutes. So we can continuously reproduce and go to the next generation fastly. It is because it is having less generation time in minutes, not in hours and days and months. And it is easy to culture. Genetic manipulation is also easy because of simple genome. What is the genome of E. coli? Like how is the genetic constitution of E. coli is there? Only single circular DNA will be there, double stranded. Okay, so because of its simple genome, will be easy to manipulate, and some fundamental biological processes are similar to the higher organisms. Then bacteriophages, they are also having single stranded or double stranded DNA or RNA. In insects, Drosophila, especially Drosophila melanogaster, it is having short life cycle. Differentiable, observable characters are present. Morphologically, if we see, for example, eye color, wing characteristics, body shape, body color, etc. And then generation time is just 2 weeks, as it is having short life cycle. Then large number of offsprings will be produced for one cycle. Easy to maintain, no, it is not harmful, and less resources are required to culture these insects. Large chromosomes are present so as to analyze at different stages of the cell division. Then several naturally occurring mutants are present, means like for in most of the other insects, what we do to study the function of a particular gene or to know about a particular character, we through the use of mutagens, we will first induce mutations and we will study about the what are the variations occurring in that particular character. But in case of Drosophila, several naturally occurring mutants are already present. We can just take them as an example to study the basic phenomena of different morphological and genetic characteristics. Then the studying on Drosophila is comparatively inexpensive. In plants, Arabidopsis and maize, Arabidopsis thaliana, it is having short life cycle. It is having small amount of genetic material. Small amount means how much is present. We shall see next slide. And clear and distinguishable traits are there in Arabidopsis to study. In maize, the genetic manipulation is easy. Why genetic manipulation is easy in maize? Means in comparison to other crops, if we see, because it is monoecious. Means its male and female organs are present on different parts of a plant. Here cob will be present, here tassel will be present. If we want to do emasculation, directly we can remove the tassel. There is no need to remove each and every anther. For example, in case of sorghum, inside one flower, so many anthers will be there. Along with that, stigma will be there. So removing of those anthers in, for example, in sorghum will be different. Whereas in maize, the tassel, that is male part, will be at the top of the plant. We can directly remove if we want to emasculate. That's why maize genetic manipulation is easy. And it is rich genetic diversity and different grain characteristics. They are easy to identify and distinguishable with reference to grain. And in animals, mice. So for model organisms, it is important for you to remember what are all the model organisms and their chromosome numbers as well. We saw that we know how many chromosomes are present in maize. How many chromosomes are present in maize?
24.
It's 20. It means in diploid condition, 20. 24, they are present in rice. Okay. So bacteriophages and mice. For maize, in diploid condition, 20 chromosomes are there. Here we studied that in Arabidopsis, small amount of genetic material is present. So small amount means here also in 2n is only 10, means haploidy is only 5 in Arabidopsis. In E. coli, we know single circular chromosome of double stranded. In bacteriophage, it's not the circular, but the linear DNA or RNA, which will be double stranded or single stranded, will be present. In mice, if we take mice in diploidy, it is 40. Whereas rats, they are having 42. So don't get confused. Don't get confused between rats and mice. This until now we studied the different model organisms also. Now we shall go to the different theories of evolution. So if you want, I'll give you 5 minutes break, or else we can continue.
Ma'am, break, ma'am.
Okay, how many, like for what, how much time you want?
5 minutes only, ma'am.
Okay, we'll be back. Now it's 10:09. We'll 10:10. We'll be back by 10:15. Okay.
Okay, ma'am.
Huh. Hope my screen is visible.
Yes, ma'am.
Okay.
Ma'am.
So welcome back after the break. Now we shall start different theories of evolution. Here we will be yes. Any doubt?
No, ma'am.
Okay. So we shall continue. Here we will be discussing different theories of evolution like spontaneous generation, pre-formation, epigenesis, inheritance of acquired characters, organic evolution, germplasm theory, and so many other theories. So first theory of spontaneous generation. Its concept was given by Aristotle. What it states is that living organisms, they originated from non-living materials, which is known as spontaneous generation. But many other scientists, they tried to disprove it, like Reddy, Spallanzani, and Louis Pasteur. And conclusively, who disproved this theory? Tyndall and Louis Pasteur. How they disproved the theory of spontaneous generation is that they conducted an experiment by boiling the organic matter in narrow mouth flask for sufficiently long time, which was tightly plugged in by cotton so that no other microbes will come inside. And in that way, after keeping that organic matter for so many days also, it did not get spoiled. So this experiment, it proved that the living organisms, they did not originate from non-living material. As that flask was covered with cotton, no other microorganisms or other like microbes from the air could come inside the flask, and it did not get spoiled. So conclusively, who disproved this spontaneous generation? Tyndall and Pasteur. Then next is reproduction in plants. This is not a theory, but a collection of different concepts regarding the reproduction in plants as well as in animals. In 1673, D. Graff, he observed the tiny vesicles on the surface of mammalian ovaries. Those were eggs. And in 1785, Spallanzani, what he proved? He proved the presence of sperm. It was essential for the development of progeny. In 1875, O. Hertwig, he conclusively proved for the fusion of sperm and egg in sea urchin. And in 1694, Grew, he described the stamens in flowers. Stamens are the male organs in flowers. Then in 1717, Thomas Fairchild, he produced Fairchild's mule. We know in 1717 what he did, Thomas Fairchild. He crossed Sweet William and Carnation, that is Dianthus caryophyllus and Dianthus barbatus. So whatever the hybrid was produced, it was regarded as Fairchild's mule. And in 1823, Amici, he observed the entry of pollen tube into the ovule. In 1884, Strasburger, he described the fertilization in angiosperms and noted the fusion of egg and sperm in nuclei. Then next theory is theory of pre-formation. Pre means like from earlier, in early only it will get formed. So what Aristotle earlier, he believed that the progeny is derived from its form from the semen, while the substance nutrition was only provided by the females. So in this pre-formation theory, we will be seeing the two different scientist groups with two different beliefs. One who believed the concept of Aristotle are regarded as animal culists. Why? Because they believe the progeny derived their form from semen and only nutrition was provided by females. And another group of scientists who believed the concept of Harvey, who speculated that progeny developed from egg cell and that semen only produced stimulatory effects called aura seminalis. This concept was further elaborated by scientists called ovists. So, in the theory of preformation, two different group of scientists are there: animal culists and ovists. Okay. Collectively, these both scientists, what collectively they believed as preformation means From the beginning only, this theory thought that a miniature human figure called homunculus is present in the sperm. Then next theory is theory of epigenesis. It was given by Wolff. He believed that adult tissues originated completely de novo. De novo means afresh. His famous successor, von Baer, he proposed that adult tissues originated completely de novo. They developed through a sequential modification, that is differentiation of adult tissues themselves. So this concept is universally accepted view of origin of adult organs and tissues from zygotes. Next theory is theory of inheritance of acquired characters. Right? Okay. So for example, in epigenesis, if we see ovum and sperm fuses to form zygote. Zygote undergo differentiation to form mass of identical cells called callus, which undergo again differentiation to form embryos and other organs, tissues, which will be developed as a particular individual. So this is what the theory of epigenesis explains. Then inheritance of acquired characters. This theory was given by Jean-Baptiste Lamarck. It was originally proposed by Hippocrates in 400 BC, but was elaborated by Lamarck. That's why it is also known as Lamarckism. Okay. What do you mean by acquired characters? Acquired characters are the characters which are produced due to the effect of environment. If for a development of a particular character, environment is influencing that character, then it is known as acquired character. These characters acquired by individuals of one generation, they are transmitted to the next generation as well. This was the belief of Lamarck, but later it got disproved. Charles Darwin, he also supported the inheritance of acquired character. And one thing, if you are especially in history, to remember the scientist, try to remember the complete name of scientist, so that you should not get if we remember whole name, then only we can opt correctly. Even some scientist name, it is getting difference of spellings as well, like Johnson and Johansson, and even Janssen. Janssen and Janssen, what they did? They did the microscope study. They developed the microscope. Whereas this Johnson, somewhere it is mentioned like this and like this. So what he did? He gave the pure line theory. So try to remember the full names of scientists. The next theory, theory of organic evolution, was given by Charles Darwin in 1859. He gave the concept of survival of fittest and also gave the pangenesis theory. Survival of fittest, we know through natural selection, the most fit or the strongest individuals will survive and lowest will gradually get eliminated. And in the pangenesis theory, every organism, it produces identical copy of itself called gemmules or pangene. This was the belief of Charles Darwin. What he believed that gemmules or the pangenes, these are produced by each and every organs, and they will get distributed or get collected in the reproductive organs. This was like from each and every organ, from eyes, from tail, hair, skin. Every organ will produce different gemmules or pangene, which will get deposited in the reproductive organ and which will get transmitted to the progeny. This was the concept of pangenesis. But Francis Galton, who was the cousin of Charles Darwin, he only challenged this theory, and conclusively who disproved this? August Weismann. Later we shall see how Weismann disproved this theory. So germplasm theory was given by August Weismann. Changes occurring in the germplasm, they are transmitted to the next generation. Through this theory, we got to know two different concepts, somatoplasm and germplasm. Somatoplasm means which will not get inherited, and germplasm which gets inherited. In somatoplasm, only mitosis occurs, whereas in germplasm, both mitosis and meiosis occurs. Means germplasm, it refers to the reproductive organs which produces gametes. So August Weismann, he disproved the theory of pangenesis. How he disproved? We shall see here that as in pangenesis, what Charles Darwin said, that gemmules will be produced from each organ and then will be transmitted to the progeny. So he cut the tails of mice and he forwarded those mice generations up to 22 generations. But in none of the generation, tailless mice was seen. Means if the pangenesis was true, then for all those 22 generations, or at least in one of these 22 generations, sorry, is my screen visible?
Ma'am.
No,
Uh,
ma'am.
sorry. Wait.
PPT is not visible, but tabs front page is open.
Okay. Just give me a moment. Hope my screen is visible now.
Yes, ma'am.
Okay. Sorry for inconvenience. So, in germplasm theory, what August Weismann did, he cut the tails of mice and he forwarded those mice up to 22 generations. But in none of the generation, tailless mice was produced. If the pangenesis was true, then at least in one generation, tailless condition should have been appeared. But it did not get. That's why the concept of gemmules or pangene was disproved. And okay. In 1909, Castle and Phillips, they conclusively demonstrated the somatoplasm is not inheritant. Means they carried out the study of August Weismann and they conclusively proved that only the germplasm is transmitted from parents to progeny. My voice is audible, right?
Yes, ma'am, audible.
Okay. This was all about the theories of evolution. Now we shall start the history of genetics. If we through this history of genetics, we can divide into 4 different eras: pre-Mendelian, Mendelian, post-Mendelian, and molecular genetics era. Pre-Mendelian genetics era, it was until the 1864, whatever genetic studies has been carried out, those are pre-Mendelian genetics. And from the experiments of Mendel, the time he published his papers, 1866, from that time, Mendelian genetics era started up to 1900. Why up to 1900 only? Because until 1900, whatever Mendel did experiments, they did not get recognized by any of the scientists. It was only the 1900 era when the 3 different scientists, Carl Correns, Hugo de Vries, and Erich von Schermack, who rediscovered the Mendel's work in 1900, and then other brief experiments on genetics were being carried out. So post-Mendelian era is from 1900 to 1944. Then molecular genetics era after 1944. Why after 1944? Because although Griffith has done his experiments in 1928, but it was conclusively proved that DNA is only the genetic material in 1944 by Hershey and Chase experiments. So then after this, we got to know that DNA is genetic material. Then in 1953, Watson and Crick, they gave the double helix model of DNA, means to describe the structure of DNA. And then we got to know the mode of replication of DNA, that is semi-conservative. So briefly, these 4 eras are there in genetics. These notes, like year, scientist, and contribution, and about the history, has been taken from the BD Singh objective book. Backside of that book, some appendices are mentioned. So those are a kind of quick revision for whole genetics history and some other aspects as well. So in your free time, if you want to read, just take a Xerox of those 4-5 pages about the history to recall the important scientists with years and contributions. So we shall start the history in 7. Wait. In 700 BC, Babylonians and Assyrians, they pollinated the date palm to ensure the fruit set. In 400 BC, Hippocrates, he suggested the inheritance of acquired characters. As I said, Johansson and Janssen. Here, Janssen and Janssen in 1590, they assembled the first compound microscope. And William Harvey, he proposed all living things originate from eggs. De Graaf in 1657, he gave the concept that follicles in human ovary, who thought them to be egg, suggested that both parents contribute to the heredity. In 1665, Robert Hooke gave the first description of cells, that is dead cells, and wrote the book Micrographia. So here in next lecture, we shall see, when starting about the cells, we shall see that dead cells, if we are getting that who discovered cell, correct answer or choice will be Robert Hooke. But specifically, if they are asking dead cell, then also Robert Hooke. What if they are adding who discovered living cell? It was not the Robert Hooke who discovered living cell. It was Anton von Leeuwenhoek, because from the tooth scum and rainwater, he discovered the living cell bacteria. Whatever Robert Hooke observed was the cells of dead cork tissue. That's why here we should try to remember that dead cell or cell is asked, Robert Hooke discovered. Living cell was discovered by Anton von Leeuwenhoek. And in 1668, Reddy, he disproved the spontaneous generation of fly larvae. Anton von Leeuwenhoek observed sperms of mammals, including man, in 1677. In 1694, Camerarius, he carried out pollination. He said that pollination is essential for seed set in maize. And Thomas Fairchild, we discussed, he produced Fairchild's mule by crossing Carnation and Sweet William. And Paul Reuter, in these 5-6 years of their experiments, he carried out hybridization between different species of Nicotiana, that is tobacco. And Spallanzani in 1769, we know that just now we read, he disproved the spontaneous generation, and also he carried out artificial insemination in amphibians and dogs. Some of the here, whatever I am marking, those are only much important, which have been mentioned in theory books of this BD Singh also. In the last of these, like in the last slides, you can leave some of the scientists which has not been mentioned anywhere in theory book. So then Lamarck, he gave the Lamarckian concept, evolution of new species through inheritance of acquired characters. Then Knight, Gauss, and Seton, they also carried out hybridization experiments. They observed dominance in F1 and segregation in F2. Then Dutrochet, he concluded that all biological tissues are composed of cells. He was only the one who divided the cells into eukaryotic and no, it was Dougherty. Dougherty, what he did, he divided the cells into eukaryotic and prokaryotic type. Then von Baer, he gave the first accurate description of human egg. And Amici, try to remember years also. In 1830, Amici he observed the entry of pollen tube inside the ovule. Then Robert Brown, he noted the presence of nuclei in cells of Tradescantia. In 1838 to 39, these two scientists, they were doing separate studies on cells. Sheldon was doing his studies on plants. Shawn was carrying out his study in animal cells. But they together came to same conclusion that both plants and animals are made up of cells, and conclusively they proposed cell theory. Then Nageli, he postulated that cells originated from pre-existing cells. And Virchow, he proposed cell lineage theory. Whatever Shildon and Shawn has given the cell theory in 1838 to 39, Virchow has made he added one extra point in that theory, and he extended that cell theory as cell lineage theory. What he added the point to the cell theory is that omnis cellula is cellula, means cell arise only from the pre-existing cells. That we shall see later. Then in 1856, Mendel started his experiments on teas for hybridization, and in 1858, Charles Darwin and Wallace, they also represented paper in front of Linnean Society of London on the origin of species. And in 1859, Charles Darwin published the book Origin of Species. And Louis Pasteur, he conclusively disproved the spontaneous generation. We saw that spontaneous generation with the narrow mouth flask, Tyndall and Louis Pasteur, they disproved. Then in 1865, Gregor Johann Mendel, somewhere it has been mentioned 1866 also, that his papers were published as experiments in plant hybridization, and they were presented in front of Bruns Society of Natural Sciences. Yeah, here it is mentioned that it was published in the Proceedings of Society in 1866. And then like Galton, he described the scientific study of human pedigree in his book Hereditary Genius. Mischer, he isolated the nucleic acids and termed them as nuclein. And O. Hertwig, we saw that he noted the fusion of egg and sperm in sea urchins. And Strasburger described the cell division in plants. Fleming, he demonstrated the longitudinal division of chromosomes and coined the term chromatin. He coined the term mitosis also, later we shall see. Then Balbiani, he discovered giant chromosomes in salivary glands of Chironomus, but he fails to really realize that these are only the chromosomes. And Fleming, he coined the term meiosis and also discovered the Lamb-Rush chromosomes. Van Beneden, he first observed the meiosis in Ascaris. Weismann proposed germplasm theory. Strasburger, he noted the fusion of egg and sperm in angiosperms in plants. Then in 1888, Theodore Boveri described centriole, and Strasburger, he described the reduction in chromosome number during gametogenesis. Waldeyer coined the term chromosome. Who coined the term chromosome? Strasburger, who first noted the reduction in chromosome number. Who coined the term chromosome? Waldeyer. Who described the reduction in chromosome number during gametogenesis? Strasburger. Then Galton, he lays the foundation of biometry in his book Natural Inheritance. And then Ivanovsky, he discovered the first virus, tobacco mosaic virus. And Beijering called the term virus for tobacco mosaic as a self-replicating agent. Whatever, here we saw that Mischer developed, Mischer isolated as nuclein. Later, Altman, he coined the term nucleic acid for those. And in 1900, Hugo de Vries, Carl Correns, and Erich von Schermack, they independently rediscovered the Mendel's paper, and independently arrived at same conclusions as those of Mendel. William Bateson, he stressed the importance of Mendel's findings in an address to Royal Society of London. And chi-square test was developed by Pearson. Then Hugo de Vries, he coined the term mutation. Landsteiner gave human ABO blood groups. McClung discovered accessory or X chromosomes. And Walter Sutton, along with Theodore Boveri, they gave chromosomal theory of inheritance. Some of the terms of genetics, like genetics, allelomorphs, homozygote, heterozygote, F1, F2, and epistatic genes. These all terms are given by Bateson. And some other terms like gene, genotype, phenotype, these are given by Johansson. And he also proposed pure line theory. Then Farmer, he coined the term meiosis. Strasburger, he observed meiosis, but the term meiosis was coined by Farmer and Murray. And then Johansson published Elements of Genetics. Bateson and Punnett gave the first linkage in sweet pea. And the first basic idea about the multiple factor hypothesis was given by Yule, which was later came as the multiple factor hypothesis. Then Hardy and Weinberg, they gave Hardy-Weinberg law for population genetics. Inborn errors of metabolism was published by Garrott, and we saw that Castle and Phillips in 1909 here. They disproved, they proved epigenesis theory, germplasm theory by ovary transplant in Guinea pigs. What they did is, in Guinea pigs, albino is dominant and black fur color is recessive. In albino female, they transferred or transplanted black ovary and they mated with them with albino. The result should be that if the Gemule concept was true, then the progeny should be of albino color. But whatever they got the progenies, they were of black color because ovary was black means the reproductive organ was black. That's why only from this experiment we came to conclusion that only the germplasm is getting transferred from parents to the progeny. Then Nielsen Nehle, whatever concept Yule gave, they elaborated and proposed the multiple factor hypothesis. And William Bateson, he published the Mendelian principles. And Janssen's one plane theory or Casimir type theory. Thomas Hunt Morgan described sex linkage in Drosophila. And Sturtevant, student of Morgan, he further elaborated the concept of linkages and gave the first genetic map. Then Bateson published again problems in genetics. Bridges, he discovered non-disjunction of X chromosome in Drosophila. Then Muller, he discovered interference in Drosophila, and Bridges discovered aneuploidy. And again Muller, he observed balanced lethal phenomena in Drosophila, and duplications are also discovered by Bridges. Aneuploidy, duplications, then translocations, genic balance theory. These are all the contributions of C.B. Bridges. And Blacksley, Belling, and Farnham, they all observed the trisomics in Daruras stramonium. And Sturtevant, he observed the first case of inversion in Drosophila. Stadler, he did the mutation induction in maize by X-rays. Then Griffith in 1928, we know he was the first who tried to discover the genetic material, and through his experiments, the transforming principle came. Then Stern, he gave the cytological proof of crossing over in Drosophila. Crichton and McClintock, they discovered cytological basis of crossing over in maize. And then Stern, somatic crossing over in Drosophila. Dobzhansky published genetics and the origin of species. McClintock, she gave bridge breakage fusion cycle. And then Beadle and Tatum, one gene, one enzyme theory. Mather coined the term polygenes. Turner, Klinefelter, Down, they all gave their syndromes as Klinefelter syndrome. He gave in 1942. Then Oswald T. Avery, Colin MacLeod, and Maclean McCarthy, they demonstrated that DNA is the transforming principle. Whatever Griffith did in 1928, he could not conclude properly. So again, they repeated his experiments in lab, and they came to conclusion that DNA only is the genetic material. And in 1946, Orbach and Robson, they with the mutagenic effects of mustard, they developed, and Lederberg and Tatum discovered conjugation. Barbara McClintock, she discovered transposons, associators and dissociators in maize, which are called as jumping genes. Then in the same year, Charkov gave his rules regarding the base pairing in DNA. Zinder and Lederberg described transduction in Salmonella. In the same year, in 1952, Hershey Chase, what they did, they proved that DNA is the genetic material in E. coli in T2. Although in 1944, these three scientists have demonstrated that DNA only is the transforming principle, then what was the need in 1952 for these two scientists to carry out again and to prove conclusively? Because like DNA polymerase, RNA polymerase, and for proteins, proteases were used to destroy these enzymes responsible for replication. But if at all we use these enzymes also in the cells, some of the remnants will be present. Although only the DNA was the kind of thing which was getting transferred, but due to some remaining traces of RNA and protein, we could not say conclusively that universally that RNA and protein are not the genetic material. So again, once again, they carried out the experiment to prove DNA as the genetic material. This we shall see in detail later. Then in 1953, J.D. Watson and Francis H. Crick, they proposed the double helix model of DNA, and Mendel gave the fine structure of T2 bacteriophage and of gene, cistron, recon, and muton. In 1956, Korenberg, he developed in vitro synthesis of DNA. Frankel Conrad and Greer independently demonstrated that RNA is the genetic material in TMV. And Ingram, he gave one gene, one polypeptide hypothesis. Then here, Francis H. Crick, he proposed the participation of an adapter. Adapter RNA is also called as tRNA. So in protein synthesis, and determines the amino acid sequence of polypeptides. This tRNA, it determines the amino acid sequence in polypeptides during translation, was proposed by Francis H. Crick. Then Okoa, he discovered RNA polymerase. Arber, he discovered presence of restriction enzymes. He postulated. Jacob and Monod, they gave operon concept. Then there are list of some other scientists as well, like here Holiday model was given by him for crossing over, like hybrid model of recombination, which we shall study in that linkage class. Then Dupro, he gave the folded fiber model of eukaryotic chromosomes. Webbel hypothesis, we know that base pairing between mRNA codons and tRNA anticodon. This hypothesis was given by Francis H. Crick in 1966. And Okazaki fragments was given by Oka. He observed the lagging strand in DNA and called those fragments as Okazaki fragments. Then Ohno, he proposed new genes originate due to duplication followed by continued mutation in one or two copies. Likewise, until 2000, here history has been mentioned. For example, Gilbert, he coined the term introns and exons. And okay, these are not much important. Whatever has been mentioned in theory that we shall read, just take earlier whatever scientists I have mentioned, try to understand those scientists, their contribution along with years. And for example, now here in 2001, first draft human genome was sequenced. It was successfully completed. And this was all about the scientist and year and contribution. Here I have also mentioned some of the Nobel Prize winners in the field of biological science, especially in genetics. We know that Behring, he got Nobel Prize in 1901 for antiserum therapy, Landsteiner for immunology, P.H. Morgan for linkage and Drosophila genetics, and like Muller for the X-rays action of mutagenic action of X-rays, and Lederberg, Tatum, and Beadle togetherly for bacterial biochemical genetics, Beadle and Tatum for neurospora biochemical genetics, and Tatum and Lederberg for bacterial conjugation. Sanger for protein chemistry, Okowa and Korenberg for in vitro synthesis of DNA, Watson and Crick along with Wilkins for double helix model of DNA. Why along with Wilkins? Because they used the Wilkins laboratory informations to publish their paper to came to conclusion about double helix model. So again, Jacob and Monod for operon concept, and Smith, Nathans, and Arber for techniques or development of techniques utilizing restriction endonucleases for study of gene organizations. Likewise, Barbara McClintock for discovery of transposable elements in maize, and MS Brown and Goldstein for identification of low density lipoprotein receptor pathway. And okay, these are the scientists who received Nobel Prize until 2006. Here, list has been given. Now, this is the list of models, theories, or hypotheses, along with the scientists who proposed those theories. For example, cell lineage theory was proposed by Virchow, but cell theory was given by Shildon and Schwann. Here it is 1838, because first cell theory was given, then only Virchow made some changes in that cell theory. So make it as 1838 to 39. Then central dogma concept was given by Francis H. Crick, chiasma type or one plane theory by Janssen, chromosomal basis of sex determination by Wilson, chromosomal theory of inheritance by Sutton and Boveri, classical or two plane theory by Sharp, copy choice theory by Belling, dosage compensation by Muller, and double helix model of DNA, James D. Watson and Francis H. Crick, epigenesis theory by Wolff. Like this, the list goes on. The first genetic map, we saw Sturt event. Then folded fiber model, we saw Duprow. And gene balance theory of sex determination, Bridges. Chromosomal basis, Wilson. Genic balance theory of related to sex determination, Bridges. Don't get confused in these theories. Then germplasm theory by Weismann, Hardy-Weinberg law by Hardy and Weinberg, hybrid DNA models of recombination by Whitehouse and Holliday, and law of homologous series of variation by Vavillo. He also gave the origin centers of origins. Then independent assortment and segregation law by Mendel, and multiple factor hypothesis by Nielsen-Ehle, mutation theory by Hugo de Vries, and one gene, one enzyme hypothesis, Beadle and Tatum. One polypeptide hypothesis, Ingram. Operon concept, Jacob and Monod. And new genes arise only from the duplication followed by mutation, and that is Ohno. Then pangenesis theory and origin of species, they both are given by Charles Darwin. Theory of synapses by Darlington. Preformation theory, Swammerdam and Bonnet. Protoplasm theory by O. Hertwig. Pure line theory by Johansen. Replicon model, Monod and Brenner. And semi-conservative replication of chromosomes, it is Taylor. Semi-conservative replication of DNA, Michelson and Stahl. Then synaptonemal complex by Moses. Theory of organic evolution by Charles Darwin. And triplet codon was given by Gamow. And wobble hypothesis we saw, it was given by Francis H. Crick. These were some of the models and scientists who gave those models and hypotheses. Now, these orbicutes or nicknames for some of the important individual organisms or some of the molecules, what we say in genetics. For example, for Drosophila of plant kingdom, we refer it to as Arabidopsis thaliana, and Fairchild's mule with the Thomas Fairchild has developed in 1717 by crossing Sweet William and Carnation. Genetic bit is a nucleotide. Jumping genes are transposons. Lamarckism, theory of inheritance of acquired character. This phenomena is called as Lamarckism. What is man-made cereal? It is Friticale. And molecular glue, because it joins the two strands of DNA, that's why it is called as molecular glue, that is ligase, which cuts the DNA fragment, as is called as restriction enzymes. They are called as molecular scissors. Then which is natural genetic engineer? It is agrobacterium. And people's choice reaction is polymerase chain reaction. Queen of genetics, Drosophila melanogaster. Secret of life, DNA. Powerhouse of cell, mitochondria. Physical basis of life, protoplasm. Suicidal bag, lysosomes. And teminism, reverse transcription. So this was all about today's class. Try to remember all these here scientists, although it has not been asked in previous year papers, but some of the important scientists and their contribution we shall try to remember. And here the nicknames are also important. It is better, as I said earlier, to take one Xerox and keep it with you to read in your free times. Hope today's two classes are clear. If there is doubt, and then let me know. Any doubt?
No, ma'am.
Okay, I think this history portion of so many slides of history were somewhat boring. So let it be, history will be like that only. From next class onwards, we shall start the about the cell, chromosome and other things. Okay. So shall we stop here?
Yes, ma'am.
Okay. Thank you all.
Thank you.
Thank you, ma'am.
Madam.